So, in this video, we will discuss the arrangement, or you can say arrangement as a taxonomy of prokaryotes. Okay. So, as you guys all know, we have discussed the introduction of this prokaryote in the previous video. Today, I will discuss about the classification. So, the prokaryotes are classified into two different classification. Prokaryote basically classified into two different classification system. Classification system. Okay, so the classification, first one classification, the prokaryote basically the two different classification system based on the two different classification system. First one is the classification that is based of the kingdom. Okay, which kingdom? Monera. If we talk about the classification, so second classification that is based on domain. In this video we will also discuss about the domain. Okay. So if we talk about the two kingdom classification, there was a scientist whose name was Carl Linnaeus. Okay, he did the two kingdom classification. Okay, first was Animalia, that he basically described the two kingdom classification. First of all, in the very starting, Animalia and Plantae. Okay, after that we were having a scientist that was John Hack. He basically introduced a new, you can say that the kingdom that was Protista. Okay, so after that there was a scientist who basically tell the groups of Protista that I'll be telling you. There was a scientist who was Ernest Hacker, who basically classified the Protista into two groups. First one was the lower Protista and second was the higher Protista. He said lower Protista is a prokaryotes because the structure is basically very, very simple as compared to the eukaryotes. If we talk about the higher, we are having the eukaryotes like the animal cell. Okay, so there was another scientist whose name was Edward Chetton. Okay, Edward Chetton. He basically, there was a scientist whose name was the Edward Chetton. Okay, he say that he says that there is the difference between the prokaryotes and eukaryotes. He told about the Edward Chetton was a scientist who told about the two things, that there is the difference between prokaryotes and eukaryotes. Eukaryotes, because they are having the true nucleus and they are having a like nucleus, and the scientists basically name this eukaryote and prokaryote as the two empires. This is a very important MCQs. Two empires. Okay, there was a Copeland scientist who introduced the fourth one classification. First one, Animalia, Plantae, Protista, and he describes a fourth one kingdom classification, which was Monera. Okay, after that there was a you can say that the scientist which name was Robert Whittaker. He basically, you can say that he did the five kingdom classification: Plantae, Animalia, Protista, Monera, and fifth one he told about the fungi. Okay, after that there was a scientist whose name was Margulis and Schwartz. They basically modified the Whittaker classification. Okay, and he basically did the Protista as a Protoctista. Okay, so there's a Monera. So in Monera, the prokaryotes were placed in the Monera kingdom. Okay. So they were placed as a, there was also assigned a separate kingdom Monera for all the prokaryotes. Okay, so there was a scientist whose name was Voes et al. He basically gave the domain system. What is domain system? Domain system. Okay, so there is the taxonomic position of the prokaryotes as a domain bacteria and a domain archaea. What does it mean? Let me tell you guys about this. Okay, what is domain? Domain is basically the higher taxonomy, higher taxonomic rank above kingdom. There we are having the you can say that the five kingdom. So above five we are having a one more, you can say the taxonomic rank. We are having that is the domain. Okay, higher taxonomic rank above the kingdom, that was given by this one scientist Voes et al. Okay, and as well as he did the classification on the basis of the bacterial species. Why he did the classification on the bacterial species? Why he did the classification on the bacterial species? What was the, you can say, that benefit? The benefit was the biotechnology. As you guys all know, we are having the biotechnology is a basically a field in which we are you can say that producing a new substance for the benefit of our body. We are utilizing those all. So he basically classified this one, the domain in the bacterial species. Okay, because we are having the, you can see the unique features of the bacterial species too. For example, we are having a bacteria who survive on 100 centigrade. 100 centigrade, our body temperature is 75 centigrade. We cannot survive at the 100 centigrade. No, we cannot survive. Okay, but these are the type of the bacteria who survive at the 100 centigrade. There was a bacteria which name is thermophilus. Okay, so as you guys all know, we are having the benefit, that is biotechnology. If you talk about the domain, if you talk about the domain, so domain is basically classified on the three types. First is the bacteria, which we are calling as the prokaryotes. Second, we are having the bacteria archaea. Bacteria archaea. Second, we are having eukaryotes. Eukaryotes. Okay, so these basically these are basically the currently most, you can say, the three domain system which are used in the classification system of the bacteriology. Okay, so if we talk about the bacteria, we are having the prokaryotes. Okay, if we are having the bacteria archaeosis, this is basically this bacteria is having the, you can say that the characteristics of the bacteria as well as the eukaryotes. As well as there is another future, they are having some, you can say that some new features. So for that the domain basically exist or arised. The domain basically arise from the, you can say that, because of archaea. In eukaryotes we are having the four kingdoms, as you guys all know. Four kingdoms. In prokaryotes we are having the Monera, and four kingdoms, that is Protista, fungi, plantae, and animalia. So these are the five kingdom. Okay, so the prokaryotes were placed in the Monera. Okay, so this was about the term. We have one more thing. The term bacteria was basically applied on the all the microscopic. In Monera we are having the bacteria, cyanobacteria. Okay, so bacteria was basically applied to all the microscopic substance. So that we are called as a prokaryotes, and prokaryotes are basically having the lack nucleus that I told you guys in that previous video. Lack of nucleus, lack of membrane bound cell organelles. Okay, and they are basically less complicated. They are less complicated than the eukaryotes. So arrangement of the prokaryotes was in the kingdom Monera. Okay, so there was a scientist who told this about the arrangement of the prokaryotes.